Okay, so we're actually awake now, and I'm slowly waking up as well. So good morning from my side. I am going to be presenting a project in which we wanted to dive into the question of organ specificity in interoception. So what kind of spurred this on is that in recent years, we've had a lot of findings that show that we have this large-scale coupling between the different visceral rhythms, so the ones from the heart, the lungs, and the stomach, and these widespread networks in the brain. But then this kind of begs this rather fundamental question whether if we look at a brain such as this, is this a highly interoceptive brain, so it's susceptible to all of these different rhythms, or is there the possibility that we are more sensitive to some rhythms than others? And although this hasn't really been empirically tested, there's really good reason to believe that we might indeed have such organ specificity. So particularly for those three rhythms, we know that these three organs have three distinct functions that they provide within the body. We also know that their rhythms are generated in different locations. And then if we look at the frequencies or the characteristics of these rhythms, we see that those are also quite distinct timescales, ranging roughly between 1 and 20 seconds per cycle. And then this is something that we've heard also a lot about during the summer school and also yesterday as well. We know that to a large extent there are shared afferent pathways between these three organs to the brain, but we also know that there are specific organ-unique pathways that could have an influence on the brain as well. So to then go into this question about whether there might be organ-specific influences, we needed to have a key brain region where we had good reason to believe that it would be coupled to all three of these rhythms. So this is why we decided to focus on the motor system. And again, good for me to speak this day because we already had some introduction on evidence between visceral rhythms and the motor system, such as Vadim's talk, where we saw that corticospinal and cortical excitability of the motor cortex is locked to the cardiac cycle, and Ignacio's findings about gastric coupling in motor regions as well. So we had a fairly straightforward design then. So we also used single pulses of TMS over the primary motor region, and we were eliciting these motor evoked potentials in the hand muscle. And every time you give a TMS pulse, such a motor evoked potential has a different amplitude. So if at the same time you then measure these three visceral rhythms, you can ask the question whether the phase at which you're stimulating somehow contributes to the amplitude that you're seeing in a motor evoked potential. So that's also the first question that we then targeted, is do we see this phase amplitude coupling between the viscera and the motor system? And then kind of where the strength of this simple design lies is the fact that we are measuring within the same person this phase amplitude coupling, and we can quantify it using the exact same measure as well. So then you can actually see whether phase amplitude coupling is independent or dependent within the same person. So second main question is then whether we do find independent or dependent effects of the visceral rhythms. And then the last thing that's important about this design is that we measured this neuronavigation, and we wanted to account for the fact that potentially visceral rhythms are inducing some sort of body movements, body sway movements that could also have an impact on our measure. So this is something that's well known in MEG or fMRI, for example. So at each pulse, we were locking the distance between where our TMS coil was in space and the intended target site so that we could regress out this potential source of noise. So the first thing we then also did was to see whether indeed it's the case, whether coil distance predicts MEP amplitude, and that's also what we found. So what you see in this direction here simply means that the closer you are to your target, then the larger MEP is. So this is quite an intuitive finding, but it was a really large effect. So then all our main analysis were done on these MEP amplitudes where we regress out this effect that is not of interest. So then we can actually look at the impact of visceral phase on MEPs. And what you see plotted here, coupling strengths, in essence what we did is we estimated for each participant the amount of coupling that they showed in 3 different models, and then we permuted their chance-Levin coupling as well. So you can intuitively interpret coupling strength as higher value, meaning higher coupling. And what we saw was that all 3 of these visceral phases were actually influencing the amplitude of the MEPs. So if we then want to test for organ specificity, it becomes super simple because it boils down to the question, if I have these participants here that show high coupling with the cardiac rhythm? Are those the same participants that are highly coupled here in the other rhythms as well? So correlation analysis. So if you then look at correlations between these different coupling strength values, we actually found no correlation whatsoever. And we tried to give some sort of interpretation to that, so we calculated the base factor, and for all three instances, we found moderate evidence for the null hypothesis, meaning that it really seemed to be the case that these visceral rhythms were having independent phase amplitude coupling effects from one another. And just to illustrate that a little bit further, what you see plotted here are basically the highest coupling participants per rhythm. So this is the person that had highest coupling in our sample with the cardiac rhythm. And what you can see from these three examples is that it's really not the case that if you're highly coupled to one rhythm, you're also necessarily highly coupled to the other rhythm. So what we found in our sample are these independent interoceptive profiles. And then the last thing we wanted to do was a little bit more exploratory, but we've seen some examples, particularly yesterday, that when we look at the cardiac and the respiratory rhythm, there is a reason to believe that excitability might be changing in particularly phases of the cycle, right? So we have these two distinct physiological phases of organ contraction and relaxation, and we wanted to see whether that's something that was present in our data as well. So what you see plotted here is basically just these phase-binned corrected MEPs, where in each case, the first three bins correspond to systole or inspiration, and the last three bins to diastole or expiration. And the first thing you can see on the group level is that we replicate these earlier effects, where during cardiac systole, we have higher excitability, and also during the early inspiration. But we wanted to take this one step further and also look at what's the inter-individual variability that you see in effects like these. So what we have in the circular histograms here is basically a distribution of the highest phase bins. So where did people have their highest excitability? And again, on the group level, you see that most participants have their highest phase bin during cardiac systole or during the inspiration phase, but it's not the case for everyone. So we also have these participants that have different patterns. And if you think about the respiratory rhythm, which is quite long, then you also see quite a large distribution of where participants actually have this peak in their excitability. And then you could say, well, okay, fine, if you have participants here that have their highest phase bin in diastole, maybe these participants simply don't have coupling, which definitely could be the case, right? So we plot a few example participants who show what, first of all, what real-life data looks like, but also to give an idea about what coupling could look like for different participants. And you can see that participant number 8 here, this is clearly a person that's following the group pattern with excitability in systole and this early inspiration effect. But we also have participants that show very different effects, right? So if you have participant 22 here, it's really late in the inspiration that there's higher excitability, and this person shows no coupling with the cardiac rhythm whatsoever. Or this case here, where it's a participant where it looks like there's coupling, but it's a completely different pattern again. So we have this amplification of excitability in diastole and actually some sort of inhibition happening during inspiration. And just to tag back to this whole summer school part that we had, I think what's also important here is if these kind of effects are conceivable, this also says something about the statistics that we use to capture these effects. If we assume phase alignment on the group level, then obviously a participant like this would be classified as a non-coupler, which maybe is not the case. So just to wrap up, we found that the phases of all three visceral rhythms are coupled to the MEPs and that they're doing this in an independent manner from one another. So we saw these individual interoceptive profiles. And I think the implication of that is that we might have different underlying mechanisms that are actually generating these effects, which seem to be at least partially independent from one another. And here I think we've seen a couple of interesting candidate mechanisms also already yesterday. But of course, with our data set, we cannot directly prove which mechanism is at play here. And then I think it's also interesting to start looking at what it could mean that different people have different types of coupling to the organs. And I think here I also want to kind of jump on Leah's talk from yesterday, which showed that this gastric coupling maybe has some implications for mental health. And maybe somehow the way you are coupled to the different organs might also have some functional relevance or not. But this is something to be explored in the future. And with that, I would really like to thank my amazing co-authors and everyone for actually showing up this morning. Thanks.
Thanks, Tahnée. Other questions? Go ahead.
Thank you. Beautiful talk. I had a little spoiler from Alex some time ago. I was like, wow, it's an amazing study. I was wondering, is there a specific cardiorespiratory phase that will better predict MEPs? In that sense, like putting together cardiac and respiratory phase and see if together they would explain something, okay, there is higher blood flow and so higher MEPs or something similar.
Yeah, that's a really good question. I did not find a satisfying way to actually statistically test that. So the circular models that we used did not allow for any interactions. We could add circular regressors to it. So I tried to address this a little bit, for example, by running the respiration model and adding heart rate as a regressor into that, which didn't seem to explain the effects. But I think maybe Vadim had some great ideas, which is nice. So I wanted to look into this, and I struggled to find a very satisfying way to do this. Yeah.
Maybe I can briefly comment.
Yeah, of course.
There is a way to do it. So this is, of course, cross-frequency coupling, right? Between
Yes.
Breathing and so what you can do, you can calculate so-called, you can make breathing moving faster to the heartbeat, and then you will have so-called relative phase delay. So basically, you can merge two cyclic variables into one. And then you can immediately analyze it. So basically, but you need to watch out that that's not just one variable, it's two merged. And then you just do ordinary statistics like you do. That's it. It's very simple. Don't add additional variable like this. So that's, I can show you it's a couple of lines in Python or MATLAB.
Yeah, let's chat about that. That sounds super interesting.
And while I have this, may I ask a question? Yeah, so it's another elephant in the room. Yesterday we had ECG, but now, you know, in TMS community, people, of course, now there is a realization that cortical excitability is not that simple. So if you put only, I checked now, you put only first dorsus interosum, right?
Yeah.
FDI, and then you can put actually also intrinsic and extrinsic muscles and also biceps. So the point is that all of these MEPs to the same stimulation, they vary dramatically.
Yeah.
So, and now what I think would be cool if one, and what we do nowadays, if you record like 7, 8 muscles, and then there is a shared variance in MEPs. And I feel that if there is a systemic modulation of cortical excitability, then we need to look at the variance of MEPs, which have a common variance, and that's most likely will be driven by remote interactions. Because otherwise they are too mosaic, like each of the motor representational muscle is quite distinct.
Yes, yeah.
But if we have a shared variance, then I think the prediction also will be stronger.
Yeah.
Potentially. Okay.
Thanks.